Normal check, you can see my slides and everything is okay, and you can hear me well?
Yeah, perfect. Thank you.
Great. Okay. So today, this journal club, I'll be talking about a paper that I was the lead author on, and it was titled "Obesity Alters Pathology and Treatment Response in Inflammatory Disease." It asks simply a question that we're now pursuing, maybe the first study of a question we're now pursuing in my own lab. This work was done when I was a postdoc, and that is, how does nutrition affect immune responses? And we had started studying obesity because to us that was one of the more drastic consequences of eating of a particular nutrition, which is eating high-fat diet or the McDonald's diet in America. And it also has big public health relevance because it turns out, at least in the United States, within a few years, it's going to be a coin flip on whether an adult American is obese or not. So 50% will probably be obese. Americans will be obese by 2030. So I think I have a presentation that's probably will last about 25 or 30 minutes, and then maybe the remainder of the time we can have a discussion or some questions afterwards. So I think this audience maybe, it's a broad audience, scientific audience, so not everyone is immunologist. So I just had a few slides to just give high levels about what the immune system is and the relevancy to this topic. So for us, the immune system, and I think for everybody, the immune system is simply a system that defends against challenges to homeostasis. And for a long time, those challenges were thought to be restricted to microbes, such as bacteria or viruses. But now people understand that these challenges can be much more than that, including things like cancer. And also, the immune system can support challenges to the parenchymal functions of cells in adipose tissue or muscle or liver. And so the immune system really is made up of several cell types, and they communicate with each other in a fairly complicated manner. And so it's quite an interesting, fascinating system to study. And one thing to note about the immune system is that not all immune responses are the same. In fact, with earlier study from scientists the last couple of decades, it's become well appreciated that the immune responses are tailored to the type of challenge to homeostasis. And so what I mean by that is, first, there are different types of immune responses. And I'm keeping this very simple by focusing only on one cell type. Every immunologist probably has their own favorite cell type. I'm going to focus on the helper T cell, which is a type of CD4 positive T cell. And it turns out that this T cell which at baseline exists in a naive state, upon elicitation of some sort of challenge, it can differentiate into three distinct forms. And of course, there are more forms, but just to keep things simple, I'm mentioning three right now. And these three different forms secrete their own different cytokines, and these cytokines enact very specific functions in the immune response. And over the last 2 decades or so, maybe even 2.5 decades, there's been a lot of study about how a naive T cell goes to being either Th1 or Th2 or Th17 cell. And this has very important ramifications for the immune response and how a host or a mouse can actually, or a human being can respond to an infection or any sort of immune challenge. This has all been done mostly in vitro. The benefit of a lot of learning this in great detail was that it was able to be done in vitro, so people could really make really strong and striking advances over the last 2 decades or so. But in the last few years, we're trying to now correlate these responses with the particular immune challenges. And people have now discovered, for instance, that intracellular bacteria or viral challenges by viruses induce a Th1 immune response, whereas helminths and parasites induce a Th2 immune response, and extracellular bacteria or infection by fungi induce a Th17 response. This is, I think, now dogma, and for the most part, it is true, at least in a mouse. The question that we had was, does the nature of the host impact the types of immune response elicited? What I mean by that is that most studies correlating specific immune challenges with Th subtypes were conducted in young and lean mice. And it's clear when you look at humans that it's never so stereotyped as what we see in the mouse, that a virus doesn't just produce a Th1 or Th2 or Th1 response like it does in the mouse, we think. It produces a mixed response. And same things with parasites as well as extracellular bacteria. And so our thought was, well, we have studied basically one type of mouse or one type of human. And in reality, humanity is much more broad and diverse than a black 6 lean and young mouse. And so we asked the question, could we think about changing the host of the mouse in a particular way and see if these rules still hold up? And that was kind of the bigger question we're trying to answer. So I don't think obesity is much of an introduction to people here, but obesity does this is a review that came in the New England Journal of Medicine a couple of months ago. And it basically demonstrates that obesity is a systemic pathophysiological shift in the body. And across many different systems, it can cause major changes. And here it just describes the things that can go wrong, but I think it also physiologically also changes many parameters in the body. And here they mentioned that immunologically, it seems like it happened, there's heightened chronic inflammation, although what that exactly means isn't always clear. And so the question for us was, okay, if you're gonna, use obesity as the fundamental change in the host, may have an obese mouse versus a lean mouse, what type of immune challenge should we use? And there, of course, for the immunologist, there are many, many types and ways you challenge the immune systems of mice. And we chose atopic dermatitis for a couple of reasons. First, it does classically elicit a type 2 immune response in lean mice. So if our idea is we want to see if there's a change in obesity and the immune response, we should have a very stereotyped and well-defined known response in the lean animals. So we knew that a couple of models of atopic dermatitis do produce this classic type 2 or Th2 immune response. That was that middle cell that I was describing earlier. And the other thing was that atopic dermatitis seemed to be in humans associated with obesity, in that as you go from overweight to obese, and as you keep increasing your patient's BMI, the incidence seems to increase, and the severity also seems to increase. This paper just shows meta-analysis describing some of that data. So we said, okay, well, let's do this first experiment, and that is, let's take a lean mouse and obese mouse. This is a mouse that was been given high-fat diet for 8 weeks versus the age-matched lean mouse. Let's give it a model of atopic dermatitis and see what happens. So the model we chose to give, and we ended up trying a couple of different models, but the model I'm showing you today, and the data comes from this one model, is we provided mice with topical MC903. MC903 is a calcipotriol or a vitamin D receptor agonist. And so if you coat the mouse ear of the skin with this vitamin D receptor agonist, you induce TSLP in the keratinocytes of the skin. So TSLP is a target gene of the vitamin D receptor. You agonize vitamin D receptor and keratinocytes, you induce TSLP. And TSLP is known to be a very strong elicitant of the type 2 or Th2 immune response, which then in turn produces atopic dermatitis-like inflammation. So what happened? Well, when we gave obese mice and lean mice the same experimental atopic dermatitis, and here I'm showing you the schematic where for 70 days or 10 weeks in this case, we gave high-fat and normal diet to mice, and for 10 days in a row, we gave them MC903. It turned out obese mice had way more severe disease than lean mice. And here you can see this by looking at measuring ear thickness. So one of the benefits of doing these disease challenges on mouse ear is that the mouse ear is skinny. So you can measure the increase in ear thickness as one measurement of severity of disease by using calipers to measure that thickness. And so one thing you may notice here is that while lean animals do have an increase in ear thickness, it's nothing like in the obese animals. And in fact, this increase, which is about 3.5-fold increase, is something that was not in the review literature. I had not seen something this drastic. Usually the experimental risk controls the difference of 50% or maybe even 1x, but not 3x. So this was something to us that was kind of striking. And it's more readily seen, I think, when you look at the histology of the mouse ear. So here, just to orient you, this is the ethanol treated. So this is just the vehicle of both lean and obese. Here is a cartilage band in the ear. And on both sides are a thin dermis and epidermis. They look healthy, they look normal, they look uninflamed. When you give the lean animal MC903, there is actually some disease created. And so, excuse me, you can see expansion of dermis as well as the epidermis, but nothing like what you see in the obese animal. And so there are many features that we noticed histologically in the obese mouse, but one thing we noticed was a lot of these blue dots. So for us, these blue dots signify leukocytes or immune cells. And we want to know what kind of cells could they be in particular. And so we did focused flow cytometry, and I'm just showing you some flow plots here, but it turned out that a good percentage of them, 15 to 20%, happened to be T cells, and of those, many were CD4 positive T cells. And so here, we first looked at Th2 cells because we thought, well, this is a Th2 disease. And so if obese mice have worse disease, they must have way more Th2 cells. And so here, I'm just showing you the presence of what are likely to be Th2 cells. It turns out that there definitely is a trending increase in the obese animals, relatively lean animals, in the lesions of the mouse ears, but not quite as strikingly high as we would expect. Look, if you base it on the actual disease score and histology. So we were kind of scratching our heads, and we looked at Th1 cells. Again, we didn't really see any big difference. And we finally looked at Th17 cells, and we saw, this humongous difference and something that really weren't expecting at all, where this is supposed to be atopic dermatitis in a Th2-driven disease. And in obese animals, it seems like, there's a presence or complete increase in the Th17 cell type. And that to us was pretty striking and interesting. And so we want to validate this and look at this more carefully in a different orthogonal way. And so we chose to look at, do single-cell RNA sequencing of the CD4 positive T cell subset from the mouse ears, both in obese and lean animals, after giving them atopic dermatitis. And so this is a UMAP describing, after clustering, looking at the non-Treg regulatory T conventional cells here. And so the major cluster I think that's probably important for you to focus on is this blue cluster at the bottom, which is the Th17 cluster. And this is, we describe a Th17 cluster, and this is the beige one, which is Th2 cluster. So one thing we noticed when we're looking at this Th17 cluster a little more deeply, more carefully, is that the RO gamma T, which is the master transcription factor of Th17 cells, seems to be seen throughout this cluster, whereas the cytokines, IL-17A, F, and IL-22, and also the receptor, IL-23 receptor, which are markers of further differentiation, or late differentiation, or late stage differentiation of Th17 cells, seem to be on the right-hand side. So if you were to further cluster these, we could have, and we were able to, we could say this is an early Th17, late Th17s. Of course, these are all done and taken in one time point. So these are just, I guess, in silico analyses. What was interesting to us is that when you look at this, when you look at the heat maps of where the CD4 positive cells from the lean and obese lie on this UMAP, we noticed that there was a preponderance of cells, the Th17 cells in the obese mice, that were in this Th17 late cluster area relative to the lean mice, which were not in this area. And so this also made sense to us because it seemed like there were maybe more Th17 cells in the obese animals, but in this case, there were definitely, all of them were expressing the cytokines, whereas the lean animals, the Th17 cells existed, didn't really express any cytokines at all. So this made sense to us, but why we want to see cytokines in the by flow, which is looking at protein, whereas here and the lean we were not able to. So this suggested to us, yes, that obese animals seem to have this increased expression, perhaps increased Th17 immune response. Is there any consequence to this? Why does this matter? It's a cool, interesting finding, but can we take it further and see that any clinical, perhaps any preclinical proof principle for a larger clinical idea? And one of the thoughts we had was that one of the great advances, achievements of immunology in the last few years has been the development of these targeted immunotherapies, in which if you have a particular disease that is known to be driven by particular types of cytokines, we're able to now provide the patient with blocking antibodies against that cytokine or the cytokine receptor. And that's tended to, in many cases, not all cases, but have really great effects in terms of therapy. And this is known for atopic dermatitis as well as asthma, where you can give the clinical version of blocking IL-4 and IL-13, and many patients do well when they haven't been doing a lot of other therapies. So I thought was, well, we're providing moderate to severe atopic dermatitis in mice. Let's give them the treatment that we give to patients, which is an anti-IL-4, anti-IL-13 blockade. And so what would happen? Well, in the lean animals, which are driven largely by Th2 cells, this is acid control. And when you give the anti-4 IL-13, there's definitely a reduction in ear thickness. There's nice protection. The question we had was, what happens in the obese case? And so our hypothesis originally was, well, there's still some Th2 cells inflammation in the obese animals, but maybe it's also Th17 driven. So maybe there'll be some reduction or maybe no reduction. But what surprised us was that, in fact, when you give anti-IL-13, it actually caused the mouse to do worse. So simply, there's an inbred mice. One is lean, one is obese. So they're genomically nearly identical, probably. Simply changing their physiological state from lean to obese, converted the therapy into an anti-therapy. And that to us was pretty interesting, and it's kind of an interesting idea. And so we followed this up more carefully. We did histology, and again, you can see the animal has some disease, but when you give the antibodies that block Th2 cells, Th2 effects of Th2 cytokines, there's a decrease. In the obese case, there seems to be, it's a little bit thicker to begin with, and if anything, it goes higher, becomes worse when you give the anti-Th2 blockade. One of the features we noticed histologically for those who are pathologists out there or look at histology a lot is we started seeing an interesting feature in the obese animals given anton for anti-13. And this feature was this neutrophilic pustules we started noting in the epidermis. And we noticed this over and over again. And it can be a marker of many things. We haven't quite figured out exactly what, but we thought perhaps this could be a marker of a loss of barrier function. So you have obese animals which are Th17 have predominance, and you give a anti-IL-13, which may just cause immune dysregulation and loss of barrier function, which may then maybe have some bacterial seeding that causes neutrophilic pustules to be produced. But it was just interesting finding that we thought that seemed to just show up over and over again in obese animals ever given this. That didn't exist the other three. So as a mini summary, obesity converts a Th2 cord inflammatory disease into a Th17 disease, which this is an interesting finding to us at baseline. But then the second aspect was that if you use biologics that target Th2 effect cytokines, they produce divergent responses depending on the status of the mouse. So it converts, what is a therapy in lean animals into an anti-therapy in obese animals. So having this set of data in the wild type animal, we wanted to probe a little bit further, more carefully, see what's going on. And maybe this is a difficult question, which is how could obesity be causing this immune type switch? And so obesity is a fairly large pathophysiological shift, metabolic shift. This is a difficult question to answer, and so we just basically started somewhere and asked, did a few different things, but we stumbled on what we think could be a plausible answer, and that is that we asked the question really, what could be happening to T cells that are in an obese mouse? How could the T cell itself become dysregulated? And for that, we early during this is data actually going back to graduate school. I had done some profiling of nuclear receptors, which are known to respond to the outside environment. And I discovered that the Th2 cells seem to be uniquely express PPAR gamma, and it's also obligate heterodimeric partner, RXR alpha. And this would seem to be unique to Th2 and not Th1 or Th17. This is all done in vitro. And further looked at it more carefully, it turns out that's true, that PPAR gamma, as in the Th2 cells, as it differentiates in vitro, the PPAR gamma levels go up in concert with its master regulator GATA3. And this is a reasonable Western blot from graduate school that the best I could do at the time, and showing that PPAR gamma is in fact expressed in Th2 cells. And this again just shows more carefully that PPAR gamma seems to be highly uniquely expressed in Th2 cells. And we were able to actually just show that we get high quality. We were curious what the direct targets of PPAR gamma were in vitro in Th2 cells. And this is simply to show that we got a very high quality ChIP-seq data. We were able to pull down the motif, the PPAR response element, which is the classic motif where PPAR gamma binds on the genome, and several target genes that showed very clean, nice peaks for PPAR gamma binding. And so with this in mind, and the thought that, well, PPAR gamma does seem to be expressed in Th2 cells really highly, we wanted to know, how is this important? How could this be useful for us? And so the reason we were thinking of PPAR gamma is that I'll show you just a few points about PPAR gamma, the background of PPAR gamma. So PPAR gamma is a member of the peroxisome proliferator activator receptor subfamily of receptors. So it was initially named because one of the isoforms promoted peroxisome proliferation. But the reason why our eyes focused on PPAR gamma was that PPAR gamma has been intensively studied in the context of adipocyte insulin sensitivity and type 2 diabetes. If you knock out PPAR gamma and a mouse survives, and there's a way to do this, otherwise usually it's embryo incognito, the mouse is born fatless. And it's known that, so PPAR gamma is the master regulator of adipocytes. It's required for adipocyte development. And if you knock out, and if you make a mouse obese, it's well known that PPAR gamma is dysregulated in adipocytes. And this dysregulation is the cause of insulin resistance or is a cause of insulin resistance in adipose tissue. So our thought was, we know that PPAR gamma does have some roles in T cell biology. Part of my work in graduate school had discovered or described PPAR gamma as being important for the accumulation of fat resident Tregs and also their function. And what we thought was, well, maybe just like adipocytes, PPAR gamma in adipocytes is dysregulated in obesity. Maybe in obesity, PPAR gamma in Th2 cells is dysregulated, and dysregulation is the cause of this type switching in the ears of mice that are obese. So one of the practical aspects of PPAR gamma biology is that though we don't know how PPAR gamma presumably has an endogenous ligand, and that ligand will then is what the PPAR gamma is sensing to either enact or not enact. It's a program, genetic gene program. We have great high affinity synthetic PPAR gamma ligands that have been generated. And in fact, we use them in basic science or research. We can also use them in the clinic. And so there have been a couple that have been approved, FDA approved, that are used as anti-diabetes drugs because they reinforce PPAR gamma action in adipocytes. So our thought was, maybe PPAR gamma is dysregulated in obesity and in Th2 cells, and let's see if that's actually the case. So the way we want to end up doing this is, again, just a analyses that relies heavily on bioinformatics and single cell RNA sequencing combined with ChIP-seq. So the idea basically was that we know the direct targets of PPAR gamma in Th2 cells based on our in vitro ChIP-seq of Th2 cells. We have a list, let's say hundreds of genes. We then ask the question, are these genes up or down in the different subsets? And do they go up or down when you look at lean versus obese? And so this is a PPAR gamma activity score that we created from this UMAP. And it turns out that PPAR gamma activity seems to be highest in Th2 cells, which will probably be more readily seen in this violin plot. These violin plots where the orange are the lean animals across the different lean CD4 cells across the different clusters. And you'll hopefully notice that the Th2 cluster seems to have the highest PPAR activity score. And what also you may hopefully notice that in the obese CD4 up Th2 cells, there's a marked decrease. And so to us, this was enough. PPAR gamma is expressed in Th2 cells. It seems to be the activity seems to be high in Th2 cells in vivo from our single RNA sequencing combined with ChIP-seq, and the activity score seems to decrease in obesity. This is enough for us to think about looking at this more carefully in a serious manner. So we decided to do the genetic experiment, mouse genetic experiment, which is if you think PPAR gamma dysregulation is important in causing this effect in obesity, let's knock out PPAR gamma in the T cells in mice, which is one way of completely dysregulating or causing loss of function of PPAR gamma in T cells. And so we had this mouse, the CD4 PPAR gamma flox, and in the lean condition, we just gave him atopic dermatitis. And again, we started noticing, yes, in fact, the PPAR gamma knockouts seem to have more inflammation more ear thickness, increasing compared to the controls, which is a CD4 alone. And this is, I think, most rarely appreciated in by histology, where we notice that these are the controls and these are the knockouts. This is all a normal chow, that the ears at baseline look okay, look normal. The controls do have increased ear thickness when you give them SN93, but nothing like in the PPAR gamma knockout case. And specifically, we thought this looks very much like, if you can remember, the obese wild type animal, where it's much greater thickness. The epidermis is way more expanded, or the dermis is way more expanded, and all these blue dots, similar to what we saw earlier. And these blue dots were again leukocytes, immune cells. And when we did and start to ask the same questions in the obese animal, we start seeing similar things. There are nuances. It's not always exactly the same. But again, when we ask the question, are there more Th17 cells in the PPAR gamma knockouts? In fact, by flow, we were able to see that there were more Th17 cells. By looking at the single RNA sequencing of the CD4 cells from the ears, what we also noticed that while the control animal, the CD4 cells in the control animal seem to be, nicely overlaying the Th2 cluster, the CD4 cells from the PPAR gamma knockout animal seem to be everywhere but the Th2 cluster. So they're in the Th17 late area, the Th1 area just right here, and also the naive-like area. So this suggested to us that it's not quite the exact phenocopy, but there's large parts that overlap. It seems like PPAR gamma is important to maintain a productive Th2 immune response. If you lose PPAR gamma in T cells, there's loss of Th2 predominance in favor of every other cell type but the Th2 cells. So we again asked the same question we had earlier, which is there a consequence to this? And so we gave anti-4, anti-13 blockade to both the controls and the knockouts. And again, in the controls, we notice a nice protection, but when you give the anti-4, 13 to the PPAR knockout animals, which don't have Th2 predominance, there seems to be an increase in the disease course, converting a therapy into an anti-therapy. And here I'll just show you again, similar histology describes it. In the control, there's nice protection, but in the knockout, when you give anti-4, 13, there's no protection. And again, we noticed, again, which is to us very striking, this neutrophilic pustule that showed up in the animals PPAR knockouts given anti-4, 13, which seemed to actually show a new feature histologically that doesn't exist in any other group. So the model that we ended up thinking about was that maybe PPAR gamma is important to maintain a Th2 focus in an immune response. And this is an idea. What does it really mean? We're not clear how this could possibly be working, although we obviously think that it works by somehow PPAR gamma within the T cell itself. But the idea being, when the PPAR gamma is sufficient animal, if you ask for a Th2 elicitant, there is, excuse me, I may be getting some allergies as well, I think, or maybe a cold. You'll get a PPAR gamma, you'll get a Th2 focused immune response in a disease that classically requires or elicits a Th2 immune response. But in a PPAR gamma deficient setting, you may end up getting unfocused immune response. In this case, we show Th17, but our data also suggests some Th1 immune responses as well. So as I mentioned earlier about PPAR gamma, when I was entering PPAR gamma, there is a great FDA-approved drug that is a potent PPAR gamma agonist. It's called rosiglitazone. There's also a partner called pioglitazone, and this drug is very high affinity PPAR gamma agonist. And the benefit of this is that we can if we believe that PPAR gamma is dysregulated in any given tissue type, we can actually usually be able to enforce activation of PPAR gamma or reinforce PPAR gamma programs by giving this drug in diet. And so we thought, well, if we believe that PPAR gamma is dysregulated in obesity, let's give obese mice rosiglitazone and see if that can prevent this type switching from type 2, type 3, or Th2, Th17 in atopic dermatitis. And so here I'm showing you a second UMAP of a second single starting sequence experiment where we took all obese animals that were either given rosiglitazone or not, or controlled, just vehicle, or that were either PPAR gamma sufficient or not in the T cells. So there are 4 groups. And here again is a UMAP, looks similar to the first one, but again, focus your attention on the bottom cluster, which is the Th17 cluster, and also this cluster, this dark green cluster, which is the Th2 cluster here. And the idea was, what happens to, in those obese animals when you give rosiglitazone? So if you look at the control, which are the control animals, you will notice the CD4 cells seem to be spread, distributed, widely throughout this entire area, except maybe not the Th17 later cluster, but they definitely have some Th17 cells. When you give rosiglitazone, though, it seems like there's a marked loss of these Th17 cells here in favor of maybe pushing the sort of the density over to this area. And so here it's better, you can see it this way, that this little foot that you can see in the DMSO or diet, it's gone when you give rosiglitazone to the control animals. But interestingly, this effect requires PPAR gamma in the T cells, because in the PPAR gamma knockouts, there's not a distribution in obese animals. Here, it seems like most of the cells are in this Th17 area. And when you give rosiglitazone, yes, there seems to be some movement towards this area, but there's still a preponderance of cells in this Th17 late area. And so to us, it suggests that if you activate PPAR gamma, you can restrict non-Th differentiation in an immune response, which we thought was exciting and interesting, because maybe you can use rosiglitazone now to create a Th2 immune response in an obese animal, and maybe then you could give anti-4, 13 as a drug, and that'd be on target, it would work. So that's exactly what we tried. We gave rosiglitazone to obese animals and asked, does anti-4, 13 treatment work? And so again, in the DMS treated obese animals, it does not work. It's an anti-therapy. If you give rosiglitazone, presumably they now have less Th17 cells, more Th2 cells, and when you give anti-4, 13, it seems to show clear protection. So here's histology of what we saw. So here's the obese animal. you give it anti-alpha-1 blockade or Th2 blockade, and you don't see really an increase. I think you see more worsened disease. With Rosi, when you give anti-alpha-1 you see nice protection. And again, we started noticing this interesting pathological feature in the DMSO fed of the vehicle, non-Rosi fed animals with anti-alpha-1. We started noticing these clear neutrophilic pustules. So this was the model that we came up with at the end of the paper, which is that there are perhaps two types of etiologies for this particular type of disease, atopic dermatitis. It could be a Th2 predominant version and a Th17 predominant version. And depending on the physical context of the mouse, you'll get either this version or this version. And so in the lean case, you get a Th2 predominant version. In the obese case, you shift the mouse to obesity and also then therefore produce a Th17 predominant version. The important consequence of this is that if you give a anti-Th2 blockade to a lean animals, you get protection because it's actually blocking these cytokines. Whereas if you give anti-alpha-1 to the obese animal, you end up with exacerbation of disease, presumably because you get rid of all the, we don't know, we think you get rid of a lot of the Th2 cells and allow these Th17 cells to go unchecked, because usually these different Th subtypes kind of regulate each other. And the interesting thing is that if you give PPAR gamma agonists, you can keep the mouse still obese, it's still obese, but immunologically it starts looking like a lean mouse and having Th2 immune response again. And therefore, when you give this drug, it'll protect the mouse from this disease. So larger ideas for in future directions. The first idea that we're pushing, this is a theme of our lab right now, our small lab, is trying to demonstrate and identify the rules to show how the obese state changes immune responses and what the rules are and how it could be working. So we're trying different types of immune challenges and looking more carefully at this particular model as well. And we also, from a part of my training was as a doctor, as a physician, and we talk a lot about precision medicine. But a lot of the precision medicine, I think, focuses on the genome. And here, I think it's useful to think about the idea that maybe our metabolic state also could be used as a parameter to think of precision medicine, and thinking about giving prognoses and also making decisions about therapy. And then finally, one of the ideas that maybe it's far off. It's an idea that only made sense in the preclinical setting, but maybe it's something to think about in the future. Is that drugs are created usually with a sometimes homogeneous population of patients, and then once it's approved, we use them worldwide, and sometimes they don't work as well. And so what if we could have these drugs that could pharmacologically, PPAR gamma defattens the obese mouse immunologically? What if we could create other drug types that maybe could be used to make a human immunologically lean or immunologically old or immunologically young? Excuse me, I have allergies coming up again. So can we just have other states besides lean and obese? And so that's something that we're thinking about as we move forward. So with that, I would like to acknowledge my advisors. This idea started in graduate school, and it kind of with Ye and Ron at the Salk Institute, but I took it to Alex Mars's lab when I was resident at the UCSF, and so this really had many cooks in the kitchen, and it's very nice to we had a great collaboration with many people working, and I'm very thankful for all of their advice and insights. But now it's also been a pleasure that I've been able to start my own lab, and here during COVID, we still keep masks on San Francisco. So we had a chance to have some fun. And with that, I'll take any questions.
Thank you so much, Dr. Bapat. I have a question. I was reading the supplementary figures of your concerning the T-Rex, and I was rather disappointed to find out that when you designed these mice, these knockout conditional mice, to delete the expression of PPAR gamma only in the FOXP3 population, that you didn't find like any differences with the obese mice. I was hoping that it could maybe be something similar to what happens in the adipose tissue, where we know that PPAR gamma is very essential for the accumulation and the function and the phenotype of T-Rex. What are your thoughts on that?
Yes, so exactly. So PPAR gamma seems to be important for the development of Tregs and fat. an accumulation of Tregs in fat. And so we had wondered whether PPAR gamma could be important for the accumulation or the importance of Tregs in the skin. And there've been a couple papers that have not focused on PPAR gamma in Tregs in the skin, but have demonstrated just gene expression levels. And there seems to be not high levels, but like medium levels of PPAR gamma expression in Tregs in the skin, but not as high as the expression in fat. And so we didn't know what to think. We thought, well, let's just knock it out and see what happens. It seems like, we did the basic experiment, which is to see if there's any difference in ear disease. And there doesn't seem to be much of a difference. But if you look carefully, I think at the cell numbers and things, I think there may have been some shifts, but we didn't follow up to really confirm, what could be, whether there could be any big differences or not. But the basic for us is if you knock out PPAR gamma in Tregs, do we see a difference in ear disease? And we didn't see one, so we kind of left it at that. But I'm sure if there's probably there's dermal fat in skin, and so we had thought, well, maybe there could be some possibly importance for gamma and Tregs, and it'd be fun to know if there is.
Yeah, I think it would be exciting to pursue that.
Yes.
And another question, I noticed that also in the supplementary figures, you compared the obesity generation. One model was with the high-fat diet, and the other were with mice that lacked the leptin receptor. I was wondering if you measured the PPAR gamma activity in those mice also in the OBOB, thinking about the differences that you saw when you fed those mice also with the high-fat diet, and you saw that there was a higher severity in the disease with the diet and not only with the obesity.
Yes. So the question you're asking is, do we look at PPAR gamma activity of the T cells in the OBOB model? And so we did not do that. But the reason we did those experiments was because we wanted to try to understand whether, when you make a mouse obese with high-fat diet and you see an effect, a very common question and an important question is the effect due to high-fat diet, just consuming high-fat diet all the time, or is it due to the fact that the mouse has now increased adiposity? And so we tried to figure this out, and that we used an OBOB model because OBOB model, they become fat on just lean normal diet. They don't have a satiety response. So it turned out that the result was that yes, just OBOB mice that are become fat on normal diet do also have increased ear disease. But if you give them high-fat diet on top of the normal diet, they get even increased ear disease. And so the answer, basically to us, that means obesity matters, but also high-fat diet probably also matters, and they both are important for causing this phenomenon that we're seeing.
Okay, thank you. Eli, do you want to pose your question?
Yes, thank you. Can you hear me?
Yes, perfect.
Thank you, doctor, for such an interesting talk. I have a question about conditional MAGAs, but in dendritic cells. Have you ever worked with conditional knockout mice for dendritic cells of PPAR gamma receptor? Because I think at least in some inflammatory responses, for example, in the lung, the dendritic cells play a key role in the type 2 immune response, right?
Yes, that's true. We focused our attentions on the T cells, and that means we didn't look at the dendritic cells. But one of the hypotheses we have going forward is presumably TH differentiation is guided by dendritic cells, and so it could be the case that maybe the action, the source of this change could be occurring in the dendritic cells. Maybe there are different populations of DCs that could be that are skewed or changed in obesity. It could therefore then skew immune responses in a particular way. So we haven't looked at it, but we're planning to look at it. Just haven't gotten to it yet.
Thank you.
Okay, thank you, Eli. We have a couple of questions in the chat. The first one is from Liz Bradstreet. Sorry. Great talk. Do innate immune cells also play a role here? Do obese mice show different profiles of innate immunity? In other words, are T-cell deficient obese mice more susceptible to AD?
Do innate immune? Certainly, innate immune cells play a role in the atopic dermatitis disease. We've started to now look more carefully and do holistically profile immune cells quantitatively. We don't know the answer to that question as to what other cell types are changing beyond the T cell subtypes. And then the question of whether T cell deficient obese mice. So we have not done that experiment. Right now we're that's important. That's an interesting question. We just haven't done that experiment to know the answer. I think those experiments, at least my experience, when you do have use RAG knockouts, or if you use T cell deficient and knockout models, there's compensation sometimes with the cell types that other cell types like ILCs. So it's not always as clean as just knocking out a cell type and being like, oh, that cell type is gone, and therefore there's no function. And so it's something worth pursuing, but we haven't done it yet. Good question.
Thank you. Elva Zenaida wants to know if it's possible to guess food bioactives that regulate PPAR gamma and will also prevent and ameliorate dermatitis disease related to type 2 diabetes.
So this is a great question. I think this is a larger question of, can we think about nutrition in a much more clinical way, and actually tell patients, we have a lot of, we eat all the time, and we have many different ways of cooking food, and food is a big part of human culture. We don't really have great, I think, guidelines or guidance from a physician's perspective of what to do for a patient who has a particular disease. We give drugs which work, but not too much about food. And so one idea is, what about are there particular foods you can eat that maybe activate PPAR gamma? And that would be great if we knew really the endogenous ligands for PPAR gamma, and we just don't. People like to say out loud that it must be fatty acids, and so if you eat high fat diets, you'll actually the PPARs. Although I don't think in practice that's been shown clearly that that's the case. So it's possible, but we are not there yet, is the answer.
Okay, thank you. And Jose Abraham Sandoval wants to know if you have tried isolating naive T cells from obese mice and differentiating them in vitro under Th17 and Th2 conditions.
A lot of people like asking that question. We have not done this experiment, and I feel like we should. The reason for not doing it earlier was that, usually speaking, when you differentiate in vitro, you give such a strong stimulus for anti-CD3 CD28 activation that the thought was maybe you're just overwhelming the, it doesn't matter what the developmental state, developmental history of the cell was. You give it cytokines and you give it such a robust activation, you won't see a difference. But I think I've been asked about this so many times that maybe we'll actually do the experiment.
Okay, thank you. And Eugenio, do you want to pose your question?
Oh yeah, thank you, Dr. Sagar. So I have a question. It's like really more specific. I was thinking of on the animal itself. It's have you looked for the levels of realm alpha as resistin-like molecule, which is, I think, is an inductor of Th17 cells and are produced highly in high-fat diet. That's why I was, I have also this question: if the T cells itself, if you transfer these T cells to any animal, like, I don't know, like rag animals, you could develop prone to inflammation. And my second question, it's more like the first one they asked before, if you have looked for CD301b dendritic cells, which are located in the skin and so are identified as regulators of differentiation of Th2 and Th17 cells.
So we are right now looking at the dendritic cell subtypes and trying to figure out if there is a baseline, if there's a difference in the dendritic cell subsets of lean and obese animals. Presumably there must be some difference that we can identify at baseline before even giving the challenge to lean and obese animals. in the immune subsets. And so we don't have the answer to that question, but we're hoping to soon discover it. And we're using flow cytometry as well as various other approaches to see if we can detect something. And the first question was the PPAR gamma. What was the question?
Like if you have looked for realm alpha, because yeah.
Yes. No, these are great questions. I was sad to say most of my answers have not done that yet. But yes, we haven't looked yet.
Thank you.
Okay, I believe that's all the questions we have for now. I don't know if you want to say anything else, Dr. Bapat?
No, these were great questions. It feels like I need to go back to the lab and do more work. Maybe after, right after I'll jump over there. But thank you for inviting me, and I hope this was fun for all of you as well.
Yeah.
Thank you so much for your time.
Thank you so much, and please follow our social networks, Facebook, Twitter, and hope to see you soon in our coming seminars. Thank you so much, and see you next time. Bye-bye.
Bye.
Close the end seminar.